Thank you very much. It's great to be here, to see many young faces. My talk title is a little bit unusual, but it really is about my life, which is science. And I think that I actually had a wonderful life in science even up till 15 or 20 years ago. So it's not something that's happened recently. Science is a really fun thing to do. Clicker. So my talk is actually going to have 6 sections. Don't worry too much about it. It's going to be historical, to some extent, how modern biology began, talking about the birth of computational structural biology. And in many ways, I'm sort of between biology and computer science, although I actually studied physics and got a Nobel Prize for chemistry. So everything is mixed up, and important to realize that. We study the motion of very large biological systems. There are also applications to human health. One of the good things about biology is it has to be relevant because we are biological creatures. And finally, some general thoughts. So how it all began. By it, I mean the fact that if you actually told somebody who doesn't know how biology works, they would think it is science fiction at a level that is hard to believe, i.e., we're alive because tiny little strings in our body are folded up in certain ways to have shapes. These strings have shapes and function, and that's how all of biology is created. Now, that is string theory to an absolute extreme. These strings also assemble themselves just to make things even more tricky. So biology works this way, and it works at a scale which is 1/10 of a nanometer, 1 angstrom scale. So it's sub-nanometer. Okay, so I think in any field, you have to always realize that you stand on the shoulders of giants. Things change because the past didn't exist before. And your past is always there. And it's not just your past, it's the past of the field. So standing on the shoulders of giants is very important. And in some ways, I was very lucky to be born in 1947, which meant I was a teenager in the 50s. And at that time, a lot of discoveries were being made to make one realize that life, biology, is really physics. One person who was really very important is a great American chemist, Linus Pauling. He actually got 2 Nobel Prizes. His second Nobel Prize was because he stood up to MacArthur, and they took his passport away. And he then became a very strong protester for freedom, not just freedom, but freedom of thought that was really disappearing in America in the 50s. His connection to these little strings I told you about that are protein, these strings are made up of a chain. And one of the ways that the chain folds on itself is to form a helical shape like this, where the chain just goes around in a spiral. The chain itself is much more complicated, but like a Chinese character, a protein is a shape. And in the shape, there are subshapes. And one of the important subshapes is the alpha helix. Another very, very important person in biology is Francis Crick. I was very lucky. I spent 2 years actually working with him in San Diego. And he, with Watson, discovered the 3-dimensional structure of DNA. DNA is a very simple structure. It's the sort of thing you see in foyers of hotels, spiral staircases. It's not a very complicated structure. What is amazing about DNA, though, is that there are these chemical groups of bases, and each group is about 30 atoms, and changing a single atom can be a lethal mutation in a human being. In fact, many lethal mutations are a single atom changing in a group like that. So you suddenly realize that biology is extremely complicated, but details really, really matter. John Kendrew was a scientist who had a very important part in my life. He, too, has a Nobel Prize. He got his Nobel Prize for solving the structure of the first protein. So you can't really see, this is actually a thread. It's a thread of chemical groups folded up on itself. This is the first protein. It's extremely complicated. While DNA is very easy to think about as a spiral staircase, this first protein would look like a pretty complicated abstract structure. You could take my cord and wrap it up. I think the thing in our real world that most matches protein structure are some of the knots, some of the celebratory knots that are tied in the Chinese culture, where you find knots going backwards and forwards. And in fact, some of these knots actually look like some kinds of proteins. But in our life, we're not really used to linear objects. It's chains folding up and doing interesting things. And all of life is based on chains folding up and doing interesting things. And then finally, Max Perutz. He was so the interesting story here is that Max Perutz was John Kendrew worked for Max Perutz, and Francis Crick was Max Perutz's PhD student. In fact, Francis Crick was Max Perutz's PhD student when he solved the structure of the double helix. But the famous paper is what's in Crick, but not what's in Crick-Perutz. So in those days, although a PhD student did an important piece of work, the supervisor who was not involved did not put his name on the work. And in fact, if you go back and find Crick's PhD thesis, in the back cover is a copy of his paper on DNA. The thesis is all about these alpha helices in the structure of hemoglobin. So this is the protein shown as in a very schematic way that carries oxygen in the blood. And it's a very clever protein because it picks up the oxygen in the lungs, but it keeps it until it gets the very tips. In other words, it doesn't release it all quickly. It releases it very, very slowly. It's designed as a very slow-release machine. This particular protein that Kendrew solved is myoglobin, which is a protein in the muscles of whales. Sperm whales die very deeply. There isn't enough space in their lungs to store oxygen, so they actually store oxygen in their muscles. But this protein actually looks very much like one of these. So the protein in us, our hemoglobin, is somewhat based on the whale protein, but is 4 copies. So this is how biology tends to reproduce things. So basically, the cycle of life that one gets from this early work is that we have information in the DNA. The DNA leads to protein structure, so you can now see this is a chain. The protein has a function by virtue of its shape. Now, almost all the things in the world around us have functions by virtue of their shapes. Knives and forks and spoons are all made of metal, but they have different functions because of their shapes. And this idea that shape leads to function is critical in biology. Now, we now have the feedback. Essentially, if this particular object, this protein, does something useful, then it'll be learned, and we learn by evolution. So in some ways, besides human learning and machine learning, we actually have, in a very, very fundamental way, biology learning. This is just to show what DNA looks like. It is basically this double helix, and the information is in this coded pattern of letters. 4 different, I guess it's a 2-bit code. I mean, each one of these is 4 possibilities. Proteins, on the other hand, are more like a chain of beads. There are 20 different amino acids, so it's a necklace of 20 different colors. But it doesn't actually look like this. It actually folds up. So it looks like, well, one thing, I'll come back to that. It looks more like this. So this is a small protein. This chain folds up to something like this, and you can see it has very chunky shape. And that shape is very precise. This scale, one of these spheres, is 0.3 nanometers across. So this is tiny, yet it has structure on the scale. The previous slide, which was out of order, just showed that these shapes self-assemble. They fold up the way they do because the energy is low in that direction. So the physics takes care of assembly. Chemistry takes care of the order of the beads. Once you have the necklace, it's a little bit like you made a car by making all the pieces, tying them together, throwing it into the air, and a folded car would come down. So it's a very, very clever manufacturing idea. Just some more pictures of proteins moving, and they move in very interesting ways. It's not too important for here. Okay, so essentially, from crystallography, we get the structure. But function depends on motion. And this is where computers came in. It's also sort of where I came in, birth of computational structural biology. So basically, again, my life is full of luck, and I think that we all have to realize that what happens to you is nothing to do with you. You are lucky or unlucky, and it's like playing cards. You deal with the deck you get, and I've been very, very lucky. So basically, I was actually born in an ex-colony like Singapore in South Africa. When I was 16, I went to England on vacation, decided to stay. And one of the reasons I think I decided to stay was that in South Africa, there was no television. And I got to England and I saw television, and it was, wow! I was completely addicted. But I was very lucky because in those days there was just one channel, BBC One, and one of the programs I had on television was John Kendrew, the Nobel laureate who solved the structure of proteins, immediately after getting his Nobel Prize, made a TV series in 1963 that explained basically this revolution in biology. And it went on every week starting on the 4th of January 1964, and this basically hooked me onto wanting to do biology. I went ahead and studied in London, did my BS degree in physics in London, and then wanted to do a PhD with John Kendrew. I wrote to him, and the answer came back saying, sorry, we have no space this year. Okay, so I was also very lucky because I had friends who were not scientists, they were business people. They were doing PhD degrees, they were doing physics first degrees, but they were really business people. And they said in business, if somebody says no, you always come back with a counter offer. So I wrote to Kendrew saying, okay, if you can't take me next year, what about the year after? And that, I think, was a very unusual thing he'd ever heard. He said, oh, then come up and be interviewed. They interviewed me, they said they liked me, but they would tell me in 1 year if they would take me in 1 year. My friend said, this is crazy, you can't accept that. You've got to go back and tell them they've got to give you an answer. You don't mind coming in a year, but you want your answer now. I went back, and basically, Kendrew said they would take me on one condition. I had to go to Israel for the year in between. Now, I had no particular desire to go to Israel, although I'm Jewish, and it was just after the 6 Days War. But Israel was a small Middle Eastern country. I would have much rather gone to Paris or San Francisco or maybe Tokyo. Anyway, he said I had to go there, and the question is why? So basically, Kendrew realized, because he had solved one of the structures, that all biomolecules are detailed, very detailed, and inside them, they look like chemistry. So the same forces that stabilize chemical molecules must stabilize proteins, and those forces must be the reason why proteins have the shape they do. It's interesting that both of these pictures come from Scientific American. In the 60s, Scientific American, which teenagers like me got in South Africa, was a key primary organ of publication for one reason. It was the only place where you had colored pictures. And obviously, if you had a structure like this, you'd want it to be a colored picture in a journal. So basically, Kendrew sent me to Israel to work with Shneur Lifson, a scientist there at the Weizmann Institute. This is how Shneur looked when I got there and how I looked. And basically, the reason he wanted me to go there was because Shneur had a PhD student, Aria Varshal. Aria shared the Nobel Prize with me, and Aria was working on something called the consistent force field. And what that really is a simple classical mechanical function for explaining the mechanics of molecules. You essentially treat a large molecule as a bunch of spheres connected by springs. And these springs have different properties, and those springs can account for the movement and the structure of these molecules. The trick is that in this function, there are certain unknowns. So, for example, B is the bond length that can change, just the distance between two spheres. But how stiff the bond is depends on the physics of the situation. So the idea that Lifson and Varshal had was to determine those properties from experiment by consistently calculating the properties. If there was no agreement, going back and adjusting the properties to get agreement. Today, you would call that machine learning. We called it least squares. They're basically very similar things. Okay, so basically, this energy function allowed, and I actually was the person who wrote the program that enabled one to calculate first and second derivatives of this energy function for Lifson and Varshal. They published a paper in May 1968. I got to Israel in October 1967, so it took about 6 months to write the program and get it published. And then what was I going to do? So I guess being curious, I looked around in a cupboard and found pieces to build a protein model. There was no computer graphics in those days. You actually built physical models of proteins by connecting bits of wire. These models cost about $10,000, but somebody in Israel had bought these pieces to make the model, and no one wanted to do it. So I volunteered to do it. And what was interesting is it's a lot of work, because each of these pieces, each of these atoms have to be at a certain height in a certain position using a plumb line and everything like that. And it takes about 3 weeks. And at the end of this process, you really know that particular protein. But you also sort of are sick and tired of doing it by hand. You'd like to put it into the computer. So I had the idea of just simply taking the listing we've been used to build it, give it to our typing pool. In those days, there was a typing pool for the computer. They put it onto punch cards, and then we could put this molecule into the computer and basically see if it's stable. And what we found is that with the kind of energy function that I described, the molecule is stable. So we could now understand very quickly that the whole thing was stable. Now, what's interesting is it's clear that Kendrew knew all of this. He didn't know I would do it. I had no idea I would do it. So again, life is surprising. Okay. So basically, this went on to think about what was called multiscale modeling, essentially choose the model that has the right level of complexity. And this is a very important lesson in every situation. If the model is too complicated, you can make no progress. And when it's too simple, you shouldn't be looking at it. So somewhere in between is the right level of complexity. There were a few attempts, and I'm not going to really go into this, but there was quantum mechanics, and there was simplified models. And basically, by this time, the idea came along that this could also be used to actually make molecules move. And Martin Karplus, who also shared the prize with us, did the first calculation of a protein moving. This is about 10 years after we got into the field. And this is a picture showing this protein moving. Now, what you can see is, if you look really carefully, that this is pointing up and this is pointing down. The only way you can actually look at motion is by movies. Movies are hard to make. They were really, really hard to make in the late 70s. This movie of showing the protein atoms moving, and the movement is interesting. The person who actually had the algorithm for drawing the sphere went on to be one of the founders at Pixar, just showing how backward things were. They're drawing a shaded sphere was actually quite difficult. And now it's, your iPhone would do this way better than I'm doing it here. Showing the protein moving, what the movement looks like is everything is shaking around, nobody is getting anywhere, but things are moving. And it's that movement that allows the protein to function. Okay, I'm going to skip this because it's not that relevant, and now go ahead to much larger structures, because that was a small protein. That protein had 56 amino acids, about 1,000 atoms. There are proteins that are much larger. Here is a protein called RNA polymerase, which is a protein that carries out an important biological function that I'll explain here. This is a much larger machine, and what one discovers is that small pieces of protein are put together to form much larger machines. This was work that was done by Huang Shui, who's a scientist in Hong Kong. He was my postdoc, and Daniel Silva is his PhD. And so, science is also very connected in that way. Here is the system. This is a large complex of proteins. It was solved by Roger Kornberg, a colleague at Stanford who also got a Nobel Prize. And it consists of about 40,000 atoms. But then you take these 40,000 atoms and put them into a big box of waters that you saw before, and you have about 500,000 atoms that are all moving together. It's a big calculation. And this particular machine is there to take a piece of DNA, shown here in blue and green, and on the DNA grow a strand of RNA. RNA and DNA look very similar. This is essentially the Xerox copier of the cell's library. You don't want to lend out your master copy of the genetic code. You want to lend out a Xerox copy. The red is the Xerox copy, and this is just the close-up showing how we add one letter to the machine. So this is a machine that has to make the red an accurate copy of the blue, and it does this by very carefully regulating what is the next position that grows. So this is a huge machine consisting of 35,000 atoms, whose job it is just to simply grow this system. This just shows the box again. Okay. Now, there's other kinds of motion. The most complicated machine that we know of in biology is the ribosome. Adi Yonat either has or will talk about the ribosome, I think, in her lecture here. And basically, this is a machine which is just mind-boggling in its complexity. What can we say about the machine? So here is a picture of the ribosome. The protein that I showed you in that first movie with a chain of 56 amino acids. The ribosome has a total chain length of 11,000. So it's 20 times bigger than that small protein. And some of these chains are made up of protein, and other chains are made up of RNA, which is like DNA. DNA can also be used to make shapes, not just to make information. So these are the RNAs, and these are the proteins. This is a huge machine. It actually has two parts. Now, what does this machine have to do? So here I'll show you a cartoon that I actually found online. So essentially, this is a piece of RNA. This is the message. This is the information. And this machine is basically going to allow adding, this is a chain of amino acids. So you want to go from the information in the RNA to the information in the protein. And it does this by basically providing a way in which an adapter molecule called transfer RNA comes in. The bond then forms spontaneously here, and everything moves on. So this machine is like I think it's probably like the 3D printer of life. It has a code coming in, and it prints out the proteins. Once the proteins are assembled, they fold themselves. So it isn't really a 3D printer. It's a chemical printer of chains, but it works because of self-assembly. This principle of self-assembly is something that we don't really know about in our real world. But it's remarkable. All of biology involves self-assembly. It knows how to go down the energy gradient and assemble itself. So that's a cartoon. The question is, can we simulate what we think the motion should look like? This is a picture of this very complicated system. We're now actually calculating normal modes. Normal modes are a very fundamental object in applied mathematics, essentially how an object would vibrate, if you bumped it or something like this. This vibration is very important for bridges, for airframes, for proteins. It involves solving an eigenvalue problem. So this is all very, very standard stuff with an enormous range of application. Even graphs, the eigenvalues of graphs are also very, very important. So essentially, this is just showing some of the motion. It allows me to say this molecule has two parts, a large subunit shown on the top and a small subunit shown on the bottom. In between of these, the messenger RNA actually runs. And here is where all the action takes place. Now, this is just these large-scale motions seen by the normal modes, but you start to see that motion of the two pieces is very important. Each of them is relatively rigid, but in fact, it's more complicated. What is really happening, the most important motion is the motion of this piece there. You see it moving? That particular motion is very important, and we came to focus on that. So this actually shows exactly the same thing, but here you see that this is the large subunit, this is the small subunit, and this is the head of the small subunit. So again, in this picture, you also see in orange solid line the RNA, and these are the adapter molecules. So if we now just think back to our cartoon, we now see the heavy piece, the light piece, the chain running, and the adapter molecules. But of course, each of these are not little cartoons, but very significant objects. So we're seeing this motion. Now, how did we find this motion? We basically, one of the troubles in biology all the time is there's great complexity, but details matter. So you're continually having to look at big data and find the interesting bits. And this is actually a very general problem, a very tricky problem. In some ways, deep learning has obliterated this problem because it says it doesn't really matter what the details are, does it work? And I must say, as a scientist, I use deep learning, but I do want to be able to say I know what's happening. It's not like this black box is the answer. Because in fact, all of life is a black box, and it's the answer. All of science, proteins fold. Why should we care how they fold? Well, we want to know details. And here again, the idea is, how do you find the details? We actually found this by looking at the three-dimensional structures of 400 different ribosomes and comparing them. And it's a little bit like you want to know how a person moves their arms. I'm going to need to use my arms. So let's imagine you see a picture of me doing this, another picture of me doing this. Now, how did I get from this to this? How did I get from this to this? Well, in fact, there were also pictures of me doing this, and maybe me doing this. So you need to take all these still pictures and try to put them in the right order. So again, this could be, somebody took your movie and cut it up into pieces and you had to reassemble it. And once you got them in the right places, we found that there were two extremes of the motion. It really was this going to this. And then because we saw frames in between, we realized the motion was essentially this. Now what is really happening is over here, my hand is close to my leg, and here the other hand is close to my leg. It's like a switch. So this is actually what the motion looks like, and we'll see it better on close-up. So you can see here, so this is like my leg, and these are my two hands. So here, the purple is close to the yellow, and here the red is close to the yellow. And we could write an algorithm to look for distances that are close in one shape, but far apart in another shape, and vice versa. And that then gives you these switch-like motions. And although the ribosome has 11,000 groups, and therefore 11,000 squared distances, there were only 3 distances that solved this requirement. So it's a very strong atom. So now what you see is happening is that this head is rotating. As it rotates, the messenger rotates. It then snaps back, and the reaction keeps on going. So this is actually now very easy to understand. You could actually make a little wooden model of a cog that you turn. And everything is happening by randomness. So this is basically the machine. Every single protein in the entire world is made this way. It doesn't matter whether it's a virus or, under the sea, 3 kilometers, or in a deep mine. All life uses this ribosome as its source of structure. Okay, so now let me just go on a bit and say, this is all fun stuff. Is it actually valuable? And I should actually say that as a scientist, I never even thought that was an issue. I was very lucky. I lived in a time when the application of science didn't seem to be very important, which I think is important. Suddenly, in the world, the time frame, the future has got closer. Everyone wants everything to be done next year, 2 years' time. Politicians want, I want things to work in my time. Who cares what happens afterwards? There's a very short time scale. And, in any field, short time scales are dangerous. If you go to the bank and you say, look, here's money, I want it back in a year's time, how much will you give me on my money? Well, you'd be lucky to get 0.1% nowadays in the world, or maybe 1%. But if you said to them, you can keep my money for 10 years, you'd probably get a lot more. So given more time, you get more. And I think this is also true for translational science. But here's an example that actually comes from my own work. And I've called it a repurposed success story. Strange name. So basically, I had done some work in the 80s on the structure of antibodies. Antibodies are proteins. They're proteins that are very versatile because they can actually recognize anything. And the way they recognize it, each antibody looks like a body with 3 fingers. This is all protein. And there's 2 parts, and they come together. And when they come together, they have different shapes of where the fingers are. Like that. The shape stays fixed, or like that, but a different antibody. And in your body, you have maybe 100,000 different antibodies. And they have these different, and depending on the shape, that way I would recognize something long and thin, that way I might recognize something round, but of course it's much more detailed. So this is the machine for recognition of the body. Okay. And why I call it a success story was that about 6 or 7 years ago at Stanford, I became an endowed chair. This was before my Nobel Prize. I actually got it for mathematically helping improve Stanford's rankings, but that's not important. Another story. But the dean was very happy with me. And he gave me an endowed chair in cancer research. Now, I wasn't doing cancer research, but in fact, what happened is that he gave me the endowed chair in cancer research because it was the next available endowed chair. He has a stack, and somebody had just released it. So I got the endowed chair on my way to meet the donors, and I'd already decided that I would spend the rest of my life working in cancer research. An endowed chair was a big deal because in the USA, your salary has to come from your grant. And if you get an endowed chair, your salary is actually paid for you. So it's a big deal. Anyway, I suddenly realized in thinking about this that instead of promising my future, I should actually repurpose my past. Always easier. And actually think that what I had done in the past was actually quite useful. So here are antibodies. These are the 3 fingers on each of the hands. They have different shapes. Papers had been published in the 60s in good journals, and I'm on these papers. And these papers try to show whether we could predict the way these shapes were? Could we predict what an antibody would recognize? And the answer was sort of yes, sometimes. Okay. Basically, in 1987, I moved from Israel to Stanford. And Stanford had a very strong pressure on startup companies. So part of it wasn't really a condition of me coming to Stanford. I didn't really care. But they said, OK, you have to join the startup company. And they asked me to join a startup company called Protein Design Lab. And Roger Kornberg was at Stanford, Levitt, Stan Falco. These are pretty well-known names in the field, early field. Levitt wrote a really famous textbook on biochemistry. Roger got the Nobel Prize for RNA polymerase. Stan Falco was a very important bacteriologist, and Lee Hood has been very important in protein synthesis and bioinformatics. So these people were on the board of this company. I was also there. And the company started with a very broad focus in protein engineering. In the early 80s, we learned how to make protein. We could actually chemically change the DNA so that the DNA would now have the sequence we wanted, and the bacteria would then make any protein we liked. So now we could engineer proteins, not by really doing it ourselves, but by using the cell to engineer, essentially by acting like viruses. Okay. So basically, they quickly realized that rather than do broad engineering, they should do antibody engineering. And this was a very important paper, probably their most important paper, which I'm an author, where basically the title tells everything: a humanized antibody that binds to the interleukin-2 receptor. So interleukin-2 is a protein receptor that's expressed on cancer cells. So you would like to have an antibody that binds this receptor, so you would target cancer cells. Now, how do you make the antibody? You take cancer cells with interleukin-2 receptor, inject them into mice, and the mice make antibodies. Essentially, this is what vaccines do. They make antibodies. The trouble is you can't really inject interleukin-2 into people. You can't use people like guinea pigs. So now what you do is you get antibodies from mice. Trouble is that antibodies from mice make you sick because they're foreign. So what you need to do is humanize the antibody. Okay. So the humanization is essentially done. Here is an antibody shown. I've now just shown a Pac-Man view of the antibody. These are the fingers. And basically, Greg Winter, who actually got the Nobel Prize a few months ago, he proposed that the way you do this, he was at the same Cambridge lab where I did my PhD, the Laboratory of Molecular Biology. He basically said, take a framework, take this part, the hand part from humans, and just graft onto the hand the fingers part that come from the mice. So it's essentially taking the body or the hand from people, and this is the fingers. And this was his proposal. And this was done in about 1984. 1987, I built this model for them, and they could now see that the fingers are actually very much touching the body. The red here are the fingers. They stick out, but they're also touching. And basically, Carrie Queen, the founder of Protein Design Lab, then had the idea that what you need to do is not just put the gray body with the red fingers, but also have a blue region where you make some changes as well. Now, he patented, this had been patented, this was then patented, and it turns out that it's always better to do more. But in patents, I don't say that. If you say patent, do AB, and now you say, no, you do ABC. And if ABC is better than AB, your patent wins. Anyway, they protected their patents. They basically got involved in biotech. The company Genentech adopted the patents from Protein Design Lab and used them to make the very first humanized antibodies using cancer therapy. It did very, very well. Protein Design Lab itself did fairly well in the 2000s, then sort of crashed. And in the end, right now, it still exists, but it's 7 employees living in South Lake Tahoe, and they're all lawyers. They live in South Lake Tahoe. It's the border of California and Nevada, so they pay Nevada sales tax, not Californian sales tax. And the reason is that the royalties were actually very significant. The patent expired. It expired in December 2014. But basically, the total royalties, say, in 2012, were $400 million, just for using the idea that ABC is better than AB. So in some ways, it was trivial. I mean, the amount of science was making a picture for them and letting them see this. But I can claim that I've already done cancer research, and therefore spend my life doing whatever I like. So it's a good idea. Okay, final thing is just some general thoughts. And so there's no doubt that my own field has been super pushed along by technology. Now, I would say the following. Getting a Nobel Prize actually depends on where you are when you hear the news because of the time difference. It's always announced between 11 a.m. and 12 a.m. in Sweden. You know, if you live in England, you know, it's an hour earlier. If you live, no big deal. If you live in, even if you live on the east coast of the USA, it's 5 in the morning, you're probably up. In California, it's 2 a.m. Now, you know, unless you are really very self-confident or crazy, you go to sleep, even if you think it might happen. And in my case, I heard the news at 2:16 in the morning, dropped my phone, they called back again, it was all fine. But basically, you then have a week where you basically feel like you're on very serious drugs. It's like taking 3 double espressos an hour continually. And you keep on getting asked silly questions, like what does it feel like and so on. And at one point in that week, a newspaper in the Bay Area said, well, what was the effect of technology? And the smart answer would have been Moore's Law, Wikipedia, goodbye. But I thought, wow, this is an interesting question. It just shows how my IQ had declined in that week. And basically, I decided to actually do the calculation. So I took what I knew was a computer in 1967, the one that I'd used, and I then looked at my laptop. It was a slightly earlier laptop. And basically, this is 2013, the cost had come down by about a factor of 10,000. My laptop was about 10,000 times faster than that huge computer. So the speed had gone up by 10,000 times. The memory had also gone up by 10,000 times. The huge computer, which occupied a hall, had 1 megabyte of memory. And now my laptop has 60 or whatever number of gigabytes of memory. So that's another factor of 10,000. And in size, I don't know how many laptops you could put in a hall about this size or half of this size, but let's just say 10,000. Maybe they have to be left open, there's cooling, there's things like this. So now you say, well, okay, what would happen if you applied that idea to something more common, a car? And basically, if a car, I said a new Volvo for obvious reasons, was like a computer, it would cost $3, would have a top speed of 2 million kilometers an hour, would carry 50,000 adults in comfort and park in a shoebox. Now, that's really funny for a car, but it's absolutely true for a computer. And in fact, if we did these numbers today, there would probably be a 3- or 4-fold more change because there have been massive advances since 2013 in terms of graphics processor units and so on. So this is an incredible force pushing forward. I think it's actually pushing the whole world forward. And, if you took away my iPhone, I'd rather say, take away water than take away my iPhone. I mean, at least I would die knowing what was going on. Okay, the question is what next? And basically, in the area of simulation, which is this idea of moving molecules, it started out in 1955 when Raman simulated liquid argon in a box, making them move around like you saw in that picture. Raman and Sillinger then did the same thing for liquid water. And what they found is that very simple forces give rise to complicated behavior. What next? And I kind of feel that the problem we face right now is that this simple energy function, which was great in 1957, involves the names of too many dead physicists, like Hooke and Fourier and van der Waals and Coulomb. We need to realize that there is quantum mechanics, and now we're actually working on quantum mechanical force fields that are very quick to calculate, but actually rely on quantum mechanics to find the parameters rather than an experiment. And we've done calculations on that, but I don't have time to describe. I'm currently a small group of people having fun. Generally, I've always had small groups. We're always doing very different things, and in many ways, I'm the computer programmer in the group. I program for them. In science, you're so dedicated to your work that family support is really important. Here is my late wife, my granddaughter, my mother. This picture was taken in 2013. Sadly, my mother, well, not sadly, wonderfully, my mother is still alive. She's almost 104. But my wife had a sudden stroke 2 years ago, and she's no longer with us. And at the time of the Nobel Prize, though, she did get an email from one of my friends Behind every successful man, there is a surprised wife, which I think is important. If you try to reverse that saying, behind every successful woman, there's a surprised man, it's really nasty. So it just shows that gender equality is complicated. Here's my mother. She's still looking great. My wife, probably at her finest moments, being, I guess, we went to the Oval Office with a bunch of Nobel laureates, and my wife just took over. We have a grandson who's called Barak. Maybe that word. It's in Hebrew. It's an Israeli name. Okay. But, you know, life is full of surprises. Here's a new beginning. My new wife, actually at an Ai Weiwei show in Jerusalem. There's an Ai Weiwei show at the museum here that you should probably go to see. An amazing Chinese artist. In life, take chances. Here am I hiking in Patagonia in 2012. I'm not doing my email. I'm suffering like crazy. We're going over a pass. It's really cold, and I'm just overheating. Anyway, I think I've said enough. All I wanted to end was with my advice to young people. Be passionate, be persistent, be original, and be kind and good. Thank you all for your attention.